Hello, everyone. In our previous lecture, we discussed adhesion and learned that pathogens must first attach to host tissues before infection can occur. Attachment allows microbes to resist physical removal by mucus, tears, saliva, urine flow, and other barrier defenses. However, attachment alone is often not enough to cause disease. Many pathogens must move beyond the body's surface and gain access to deeper tissues where nutrients are more abundant and immune defenses may be easier to avoid. This process is known as invasion. Invasion can occur in several ways. Some pathogens enter and survive within host cells. Others produce enzymes that break down host tissues and allow them to spread. Still, others exploit wounds, medical devices, or other breaches in normal barriers to gain access to new environments within the body. Successful invasion provides several advantages. It can give pathogens access to nutrients, allow them to spread throughout the host, and sometimes help them avoid immune defenses. At the same time, invasion often causes tissue damage that contributes directly to disease symptoms. In this lecture, we will examine the major strategies microbes use to invade host tissues and explore how these mechanisms contribute to pathogenesis and disease progression. As with before, this is not going to be comprehensive. We're just going to use a couple exemplars to go over some of the main types. By the end of this module, you should be able to differentiate major invasion strategies. Pretty straightforward. All right. So we've talked about this again, but let's revisit a little bit. The first step of infection is encounter. A pathogen must come into contact with a suitable host. This may occur through inhalation, ingestion, direct contact. That direct contact could be with surfaces, through sexual contact. There could be vectors like tick-borne diseases, those sorts of things. Contamination on medical devices, so many ways. Next comes adhesion. The pathogen must attach to host tissues and resist physical removal by mechanisms such as mucus, tears, saliva, urine flow. This was the focus of the previous lecture, of course. Today, we're going to talk about invasion. Once attached, many pathogens move beyond the body surface and gain access to these deeper tissues. And then after that, we're going to move on to evasion. How do these pathogens survive host defenses, such as phagocytosis? And then we'll talk a little bit more about damage that these cells can do during the process of invasion. And with that, let's take a look. Attachment is absolutely essential for infection because a pathogen that cannot stick to a host tissue will simply be washed away. But attachment alone is often not enough to cause disease. Attachment allows a pathogen to establish and colonize, or establish colonization, but many pathogens gain important advantages by moving beyond the surface. By invading the host tissues or entering host cells, pathogens can gain access to nutrients, as we mentioned earlier, and some can remain attached long enough that they will produce toxins that can cause damage to the host cells. And so let's go through a couple of examples of how these deeper invasions occur. First, we're going to look at this specific example of invasion using uropathogenic E. coli, or UPEC. The infection begins when bacteria from the gastrointestinal tract enter the urethra and begin descending through the bladder. At this point, urine flow presents a major challenge. Every time the bladder empties, bacteria risk being flushed from the urinary tract. To overcome this, UPEC uses type 1 pili or type 1 fimbriae. These adhesins bind receptors on the bladder epithelial cells and allow the bacteria to remain attached during urine flow. For many bacteria, attachment would be the end of the story. They would remain on the surface and continue growing. However, some UPEC go one step further. After attachment, host epithelial cells can internalize the bacteria through endocytosis. Rather than being destroyed, some bacteria survive inside these cells, and that's what we can see in this image here. Once intercellular, the bacteria gain several advantages. They are protected from urine flow. They're less accessible to immune defenses and may be partially shielded from antibiotics because they'd have to pass through the epithelial cell. Inside the epithelial cells, UPEC can replicate and form intracellular bacterial communities. Uh, in the example here, or in this example, it illustrates an important concept about invasion. The pathogen is not actively burrowing through tissue. Instead, it exploits normal host cellular process to gain entry into the protected environment. In this case, it used the cell's own process of endocytosis. Um, Once invaded into the cells, you have access to all those intracellular nutrients. You have protection from the physical removal, and you have refuge from host defenses. So let's take a look at Neisseria. Neisseria uses a different invasion strategy than UPEC. They use very similar adhesion strategies, but very different invasion strategies. Rather than simply waiting to be taken up, Neisseria actively manipulates host cells to promote its own entry. The bacterial surface contains proteins called OPA proteins. These proteins bind receptors on the host cell surface. When this interaction occurs, the host cell receives signals that something should be brought inside. Those signals trigger a rearrangement of the actin cytoskeleton, which is, as you remember, the structural framework that helps determine cell shape and movement. As actin reorganizes, the host cell membrane begins to wrap around the bacterium. Eventually, the membrane surrounds the bacterium completely, and the cell internalizes it through endocytosis. So, in this case, very similar to UPEC, but it's more active than it is passive. The important point here is that the bacterium isn't forcing its way into the membrane mechanically, just as before. Instead, it is manipulating normal host signaling pathways and in convincing the host that it should be engulfed. This strategy allows Neisseria to cross epithelial barriers and gain access to tissues that would otherwise be inaccessible. Um, Now, it does differ from UPEC because UPEC primarily benefits from being internalized after attachment, where Neisseria actively induces cellular changes that promotes that uptake. So far, we've looked at pathogens that invade by entering the host cell. Streptococcus pyogenes often takes a very different approach. Rather than relying primarily on intracellular invasion, it spreads through tissues by breaking down the structures that normally hold tissues together. To accomplish this, the bacterium produces several extracellular enzymes. Hyaluronidase degrades hyaluronic acid, an important component of connective tissue, often described as the cement between cells. Streptokinase activates plasmin, which dissolves fibrin clots that would otherwise help contain the infection. DNase breaks down extracellular DNA released from damaged host cells and neutrophils. This reduces the viscosity of infected tissues and helps bacteria move more freely. Together, these enzymes break down physical barriers that normally limit microbial spread. As tissue damage accumulates, cellular contents are released and inflammation increases. The resulting damage creates additional opportunities for bacterial growth and dissemination. In severe cases, this process can become a rapidly spreading necrotizing infection, sometimes referred to as flesh-eating disease. Notice how different this strategy is from UPEC and Neisseria. UPEC gains protection by entering epithelial cells. Neisseria manipulates host cells to be engulfed. Streptococcus pyogenes invades by degrading the extracellular matrix altogether and creating pathways through host tissues. So we've seen how pathogens can invade host tissues in several different ways. One strategy is what we call passive entry. In this case, the host cell internalizes the pathogen and the pathogen takes advantage of that opportunity. UPEC being a prime example. After attaching to bladder epithelial cells, some bacteria are endocytosed and survive within the host cell. Once inside, they gain protection from urine flow and some immune defenses. Second strategy is active entry. Here, the pathogen actively manipulates the host signaling to promote its own uptake. Neisseria uses surface proteins that interact with host receptors and trigger rearrangement of the actin cytoskeleton. The host then engulfs the bacterium because it thinks it's supposed to. And the third strategy is just Guns ablazing, barrier removal. Rather than entering cells, some pathogens spread by breaking down the structures that normally hold tissues together. Strep pyogenes produces enzymes that degrade components of the extracellular matrix, allowing the infection to move deeper into tissues. Although these mechanisms are very different, they all accomplish the same goal, giving the pathogen access to environments that improve survival. Growth and persistence within the host will then proceed. The important thing to remember is that invasion is not a single process. Different pathogens have evolved different solutions to the same problem. How to move beyond the body surface and establish a successful infection. All right, and with that, I appreciate you coming to this lecture. I'll see you in the next one.